data and digital in this themed session. I think probably with just the minute I've got to kind of introduce this stuff, it's quite difficult to draw boundaries between a lot of the themes and the presentations and stuff that we've seen at this point. If you've been up, you've seen some of the posters, you've seen some of the demonstrations. A lot of the data and the digital technologies, they kind of sit across all the work that we do at this point. And I think, as associate director, and with that kind of remit to look at that sort of transformational piece, that journey in the last 10 years has been quite remarkable. And I think, given that we had COVID as well, there's been a kind of an explosion really in the way that we're using these technologies and that we're using data to drive that in the last few years. So with the investments that Alistair's talked about, I think you can probably expect quite spectacular things in the next few years. So while I've got a mic for a few minutes here, just a big thanks to the whole team, because it's all the data and all the models that lets us do the whole digital twin thing and makes those factories work. So well done to everybody there. So to kind of get back on track, our first speaker today, we've got Rachel Smith as a senior lecturer at the University of Sheffield. Where are you, Rachel? Yeah, you're there. So Rachel's been a long-term contributor to the CMAC efforts. Has got interests in particle fluid, particle interactions, and doing all the modelling and kind of cool stuff around that. A lot of that work over the years has been kind of key to delivery of our digital twins. And again, I think if you've been in recent years, you'll have seen a lot of that work. And I say it's been a kind of keystone to some of those initial digital twins. So today, Rachel's going to be talking about linking drug product and microstructure and performance. So take it away, Rachel. Cheers.
So today I'm going to talk to you a bit about a lot of the times when we look at models, we think about a model for an isolated unit operation, for example, or we think about process chain modelling. But often we don't tend to think about the product performance of the products that we're making. So I just wanted to talk to you a bit first about some of the motivation for the work that we've been doing recently. So, as I said, we have an awful lot of models these days for understanding how will my process decisions and my material properties influence, for example, the structure or the size of or other attributes of my product. We have somewhat less of the understanding of how does my product attributes affect the performance of my product. Because actually what we really care about is not the attributes of the product, we care about the performance of the product, and that's what we actually have to keep an eye on and try and achieve. But there's a real gap in the science so far, and that gap is about how do we link these process models with the performance models? How do I run a model which gives me certain attributes of my particles, and then run a model which tells me how that product will perform? And even more, how do I use these models to start to design my process to give me the desired performance? And that's really key. And I think it's something that people are certainly starting to work in, but it's still very much an open area of research. So we started looking, and this is just an example here of a wet granulation model. You have a certain structure or certain properties that you end up having as your output, and then you come into a product model and you understand how does that product perform. When we started working on this, there's an awful lot of different performance metrics we might care about. We might care about the dissolution, disintegration of these products. We might care about the attrition resistance, the strength of the products. Stability is a really important area as well, as we know in pharmaceutical manufacturing. But we've taken a bit of a case study here, and we've decided to look at dissolution, disintegration, and in particular actually disintegration of these products as a case study to start with. So, as I said, there's been a lot more work done on the process models than there has been on the products. So we decided to start with the end, start with the product models, and try and develop some models that are able to be coupled with process models. It's not to say there aren't product performance models, but the challenge is really in trying to couple those models with the process models, right? We need that common information at the center between the two models. So we decided to start working on a modeling framework, a mechanistic model basically, for how does a granule disintegrate. We have three primary steps here. We have a liquid imbibition, so liquid going into the granule itself. We have liquid absorption into the particles inside the granule. This is particularly important because we're very much focused here on a swelling-driven breakup. Part of the reason we're doing that is because it's not been well understood so far. So we're looking at how is a liquid absorbed into the constituent particles in a granule. How does that stress build up over time and then cause the granule to disintegrate? I've left a bit of this out, so we've got all these equations. Obviously, it's a modelling paper, so we have to put some equations on there, but don't worry about them. But basically, this is a single granule model. What we're then using is a population balance to give us evolving populations of the particle size and porosity over time. So what we're doing is we're actually getting a distributed particle size, a distributed particle porosity over time in a disintegrating vessel, for example. So we've got a model. One of the first things we had to do was start looking at, well, what's important in this model and what is not so important in this model. So we looked at formulation parameters and we looked at material property parameters. Now I'm not going to go through all these different parameters in great detail. One thing I just wanted to note was that there's a lot of parameters here, right? These are complicated models, and the reason there's a lot of parameters is because a lot of things matter, right? These are complicated materials and the complicated systems, so we have a lot of parameters. The other thing I wanted you to note is just that we've got these color coded here. So if they're red, it's highly dependent. If they're green, there's a low dependency, right? And if it's orange, it's somewhere in the middle. So we also have, not only do we have a lot of parameters, but we have a lot of parameters that significantly affect the model results. So that's something to note here. So rather than showing modelling results straight up, I'm going to talk a bit about our experimental investigation and throw all our modelling and our experimental results together. So there's no point making a model if you don't test it. So we did some experiments where we generated some granules. And as I said, we were deliberately trying to look at the way granules disintegrate here. So we're not particularly trying to look at dissolution. We're looking at disintegration or dispersion models here. So we made granules, made high shear mixer granulated granules. We changed the amount of super disintegrant that we added. So SSG we used basically as our super disintegrant. And we also changed the amount of polymeric binder we added. We use HPMC as a polymeric binder basically. And here you can see that by changing the polymeric binder, we change the size distribution of our granules. So that would be captured in the process model, right? But once we've got those granules, we want to understand how do they swell and how do they break up over time. And I'm going to focus particularly on the swelling here. So we had to develop, and we've done this work in conjunction with Daniel Markle, the University of Strathclyde, and his team. We designed a flow cell in which we can put a particle, we can put a granule, and we can see how it changes over time. So we've got a flow cell, we're putting fluid flow through the cell over time. We have an optical microscope and a camera. And we also developed some data and image analysis software. We know there's loads of image analysis software on the market, but it's a real challenge to get any of them to work for one particular system, so we just went back to scratch and did it ourselves after trying a few different things. But effectively, we have a little camera, we can see the way the granule is changing over time. That means we can actually measure the swelling, and you can see up in the top left-hand corner there of that video, the diameter is changing, and bottom left-hand side is the time changing. And we can map that time, that change over time. So what this allows us to do is really, I guess, at an individual granule level, verify our model. Now, we don't think to use these models that you need to do this, right? We want to do this to make sure we believe the model works well. If you're going to apply this model in industry, there's no way you're going to put an individual granule under a camera and study that and do that a billion times, right? But this is what we're trying to do to get confidence in the model. You don't need this to parameterize it fully. And what we see here is that for our highest disintegration value, obviously, you get very rapid swelling, as you might expect, and then we get breakup. And so the curve stops when we actually achieve breakup because we no longer can measure an individual diameter anymore. As we reduce the disintegrant concentration, we reduce the rate at which the swelling occurs, and we get longer and longer curves there. So we've got our, so now I'm starting to introduce some model data. So that was just experimental data. Now we have our models here, and we've got our model as the dotted lines on these graphs, and we've got our experimental data as the solid lines on the graph. And you can also see we've got our standard deviation for the experimental data there. So one thing I want you to note is that there's some substantial difference some substantial variation in the experimental data, okay? That is to be expected. That's not error, that's a function of the heterogeneity in granules, right? So there's always a huge amount of heterogeneity in a granular population. But what we basically come from here is that to a reasonable degree, we're confident in our model. We're pretty confident in our model, and we've done this analysis on a very, very tiny analysis. There's not enough time to talk about it here today, but we've also gone ahead and validated the model for the population balance part of the model, using FBRM, for example, to look at the evolving particle size of these granules over time. But next I wanted to talk a little bit about, so I've talked about granules, right? But we need to talk a bit about tablets, because most of the time we don't eat granules, most of the time we take a tablet. There's been some combined work between Siemens PSE, between the University of Strathclyde and Imperial, to develop a brand new tablet disintegration model, basically a tablet dissolution disintegration model. Now this is not in the current release version, but it's only recently been completed. But there's an opportunity for us to use these experimental techniques that we've been looking at, and previous experimental data, to try and understand how well does this model capture swelling in tablets. So again, don't worry about the equations. Basically, we have an equation which describes the water penetration, and we also have an equation that describes the swelling of the tablet. We've got a whole load of different assumptions here, which effectively simplify the system for us. So my postdoc, Neeru Bala, has been working hard to try and understand, first of all, if we just take the model and we separate out the liquid penetration from the swelling, what is the dependency? What we do a GSA on this. What is the dependency of different parameters on the model? And we find, of course, as you might expect, porosity features very strongly in both the liquid penetration and the swelling, and that's no surprise there. By doing this, we can understand that, for example, for the liquid penetration model, the surface tension is not a huge influence. So if we don't have a massively accurate measurement for the surface tension, for example, that is not such a big drama. We then worked with some historical data from Daniel Markle's group at Strathclyde, and this is really lovely data. It's taken using terahertz imaging, and what it means is that we can actually see the liquid front in a tablet, and also the height of the tablet change over time. So this data has all been published, but and don't worry about this model, the model on the graphs. But basically, what we're using here are the data points on these graphs on the left. Effectively, we can see both the displacement, which is the liquid penetration into a tablet, and also we can see the swelling. And so this is really lovely data for Neeru to start looking at, and to use to effectively test this new model that's from GPROMS. And what we can see here is that we're getting really nice agreement between the model and the experimental data, with the exception of when we get to sort of slightly higher times, where we see that the experiments tail off the liquid penetration a little bit earlier than we expect. And we can also do this again for the looking at the swelling. So this is instead of looking at the liquid penetration into the tablets, is looking at the swelling of the tablets themselves. And as I said, we get pretty good agreement. We've done this again also for not just for the one porosity, but for lots of different porosity of tablets. Again, we largely get pretty good agreement, but with one caveat, which is when we look at the swelling, we do see this really enormous experimental variation, and again, that's a property of the distribution of performance of tablets. So it's really something that we need to keep in mind when we run a model. We are effectively getting an average, what we might expect as an average. But actually, we have to expect a huge variability in the real results. So, in summary, we have a new mechanistic model for swelling-driven dispersion. We've done some GSA on that, and to understand the influence of various parameters. And we've also done a lot of complementary experimental investigation with lots of different variation, and we're pretty happy with the model agreement. We've also been looking at tablet mechanistic disintegration of tablets, and with using some really lovely data taken from delivered by Daniel Markle. we're able to see that the new G-prompts model works pretty well. As I said, the whole ultimate aim of this, though, is to start to be able to solve that inverse problem. So we're trying to inverse engineer, reverse engineer these systems so we can design a process to give us a desired performance. So, and that work is ongoing. And I'd like to thank all the partners, all the collaborators, all the industry teams, everybody. Thank you very much.
Okay, thanks, Rachel. Good stuff. Cheers. Do we have any questions for Rachel? We've got one in the app. We're doing well. Anybody in the room? Right, we'll start with the app. So Peter Hollowell is asking, can the models predict when the granules break up?
Yes. So I wasn't able to show that today. The bit I could show I had time to show was looking at granules swell. We can predict the swelling, but we have done secondary, some further analysis where we're looking at, we put the single granule model into our population balance model, which predicts both the time that the granules break up, but also the distribution of fragments. And we have compared that with FBRM data, and we can show that we're getting pretty reasonable agreement there.
So, is there also a range of times when they?
Yeah, so we have a distribution. Effectively, we have because it's a population balance. We have distributed properties, right? And then those distributed properties, the granules, the distributed properties will break up at different times depending on their properties. So that was one of the reasons we went down the population balance route, is because it allows us to handle these distributions.
So ultimately, would it ultimately be like an ensemble
model in the end, where you would have an understanding of what properties are more likely to be there, and then you'd build up
So ideally in the end, yeah, you're right. So ideally in the end, we build up effectively a surrogate model, right? So we can use this data to build a surrogate model, which not only tells us what parameters are important and which aren't, but which also means we don't have to keep running the PBM all the time.
Thank you.
But we haven't quite got to that bit yet.
Thanks. Just quickly, when you're talking about surrogates, and those models seem quite complicated, a lot of parameters.
Yeah.
Something I guess we'll be looking to do in the next few years is to actually make a lot of these complex model chains and stuff run in real time. So, do you have a feel for the current model performance and maybe, in the future, how we can get more performance out of them, speed them up?
So the current model is a little slow, but one of the things I think, these mechanistic models, a little slow, it's just not quite real time. We're not talking days and days, it's not quite real time. But I think the real power in these models is to feed into surrogate models, right? So we basically create huge data sets by running models rather than running experiments. We then create surrogate models that are real time. We also have a really good understanding of our confidence in the models because we have so many data points. So I think we need to sort of stop worrying about trying to run mechanistic models in real time. We run mechanistic models to train our machine learning models, right? Yeah.
No, I think that's the way we're going to go about it. Any last questions for Rachel? Okay. Cheers, Rachel.